NTU World of Wisdom. Welcome to High Impact Thesis. In this podcast, we speak with researchers from various scientific fields to talk about the motivation, goal, and potential impact of their research. We also want to give you a sense of how a Ph.D. is carried out with an emphasis on the PH, the philosophical aspects involved in pursuing a Ph.D. Hello everyone, welcome to the HEAT podcast. Today our guest is Ms. Anna Korsakova, and we'll talk about her research work, what she's doing, what the implications of the research are at a high level, as well as what tools she uses for her PhD that can be useful for other people who are doing PhD here at NTU in Singapore or around the world.
Hi everyone, my name is Anya Korsakova, and I'm really pleased to be here today in this podcast, and I'll be happy to talk about my research and the implications of my research.
All right, so Anna, welcome. So we really want to get started by talking about your research. How do you explain it to a lay person who has no idea, what your field is about? They've never heard of it. How do you explain your research?
All right, so I guess I need to start from afar because I'm a last year PhD student, and I'm working in a biophysics lab at NTU SPMS, School of Physical and Mathematical Sciences. Mm-hmm. And actually, although my lab is called biophysics, my background is in theoretical physics. So it's like an intersection of physics, biology, and bioinformatics. And I have a few major projects, and I can talk about all of them later on. Mm-hmm. But recently, we published a paper on one of the projects that I'm working on, which is prediction of alternative RNA splicing with neural networks. And this is basically this is a purely bioinformatical project plus machine learning.
You want to slightly unpack that, this RNA splicing, what is it?
Yeah, sure. So RNA splicing. So the central dogma of molecular biology states that DNA is used to make RNA and RNA is used to make proteins, right? Mm-hmm. And then, because the set of genes is pretty much the same for all humans, apart from some mutations and variations. So the sequence itself is almost the same. Mm-hmm. But actually, the proteins are very, very diverse. And it means that there's a lot of processing somewhere in between. So basically, you have a gene, right, in the DNA, and then it's transcribed to RNA. And at the level of RNA, there's a lot of processing. And this processing is called alternative splicing. So from one particular gene, you can actually have a few versions of the same protein. Mm-hmm. And they will be different. Okay. And it's very important because, at different stages of the cell development, like, for instance, stem cells or young cells or senescent cells, you will need a different set of proteins. Mm. So this is the process by which you will get this different set of proteins. And because this process, because the sequence is the same, mm-hmm. So it probably depends on something else. So it depends on the auxiliary signals, like proteins that are present in the cell and other things, and that's what we're studying. Okay. Basically.
So what you're saying is that you can have the same sequence of ATCG and mm-hmm. so on, and that same sequence can produce different proteins depending on the processing that happens
on the background
you've transcribed. Yep. And so what is the motivation for this work?
So the motivation is because, well, first of all, because it's cool, and okay. It's very interesting to study what exactly affects the alternative splicing, because it's not very well studied yet. Mm-hmm. So the thing about all these processes is, imagine a cell, and you have so many molecules and so many signals in a cell, so we don't really know what affects what. We have mm-hmm. some clues, but not all the clues. So this work is mostly about the data science of the bioinformatics. So we're trying to figure out which signals, which proteins actually affect the alternative splicing of some other gene. Mm-hmm. So as an input, we'll have a sequence itself, which will be the same for, let's say, all humans, because we disregard all the mutations. And then we have auxiliary signals that are specific to a cell or to a human, to a cell type, to tissue type, and so on and so forth. Mm-hmm. And based on these auxiliary signals, we'll have different outcome for alternative splicing. Mm-hmm. So we're trying to look at it and outline which signals exactly are more important, which are less important, because these alternative splicing, these protein isoforms, are very important for cell development and cell regulation. And for instance, some diseases, like some cancers, they can be cured if you can switch the isoform, the protein isoform, back to the normal one. So if we find what exactly affects this switch in the first place,
Right.
the switch to the cancerous isoform, maybe we can revert it. So that's the motivation.
And just out of interest, do we have a figure for how many proteins the human body has, or is
So the number of genes in human genomes so far is about, if I'm not mistaken, 30,000. But given that I already mentioned that actually each gene can have different isoforms, protein isoforms, so the number of isoforms I think is about 120,000. So it's a very large number.
Okay.
And when you say like signals, like you're analyzing the signals, what are these signals and like where are they coming from?
So these signals are the protein expression levels of other proteins actually. So, it's like self-connected chain. So basically, we're studying whether the splicing of one particular gene into different isoforms will be affected by other proteins floating in the cell. Because we know for sure biologically that it will, because other proteins and other small signals like small molecules and so on and so forth also act like signals to regulate the splicing and other processes. So we just outline, we know approximately which proteins might affect this process. So we're testing which ones actually do.
Is it sort of like a feedback loop where some cells will produce proteins and these proteins will affect other cells?
Yes,
in a
exactly,
way?
exactly.
So proteins in the body are able to sense the so to say the concentration of certain other proteins.
Yes,
right?
exactly.
So then they act like sensors in other words.
Like switches, sensors, yeah,
I see,
precisely.
And you also mentioned like disease, like cancer and other. So are you targeting specific diseases or just studying the whole phenomena?
So for now, we're studying the whole phenomena, and the work that we published was mostly about the data itself, right? So we're just outlining which subsets of data can probably affect the splicing process. But right now, we're trying to make a small, maybe discovery, because it's very useful for clinical applications if you can, find a narrow case, just one case of the splicing effect. So for instance, you can find which protein affects the splicing of your gene of interest, which is related to cancer, and then maybe you can target it with drugs. So that's what we're working on right now. But this part is actually much more complicated.
Right. So now you're just targeting the splicing process, like what proteins initiates or which proteins results in this type of splicing or that type of splicing, something like that.
Yes, something like that. Subsets of proteins, just outlining.
Subsets of proteins.
Yes, narrowing down, like we have a lot and then we're just outlining which subset is the most important, and then we select a gene of interest, and then we're trying to mutate them in silico. So basically just put the inputs to 0, and then you see if the output changes, which is the splicing of your gene of interest, then probably this thing is important, and then you can test it experimentally.
Right. Okay, so that's another word I was not sure what it means when I was reading up on your work. Silico. So it comes up sometimes.
Oh yeah. So
what is that?
There are these abbreviations or phrases. How should I call them? In vivo, in vitro, and in silico. So basically, in vivo means that you're testing something in a live organism. Okay. And then in vitro means that you're testing it in glass, vitro glass, right? Okay. And in silico means that you're testing it on a computer. Yeah, silico.
Okay.
Makes sense, right?
Right. Okay, so as we communicated with you on the email, so first you start with the overall feel, what's happening, what's going on, why did you get into this?
Right.
And now in this section, we want to get to know your personal work in more detail. Like you go, are you staying on campus or?
No, right now I'm staying outside. I'm renting a room.
Okay, so you come from somewhere in Singapore. You come
Right.
to NTU every day, go to the
Right.
lab, I
Not
guess.
every day.
Almost.
That's a secret.
All right.
Okay, not really.
I mean, nowadays, yes, I can understand. Yeah. So we want to get into like, what is your daily? What are your daily tasks?
Routine, right.
Yeah, like the routine.
It actually varies a lot, and right now, actually, I'm not at NTU per se. I'm at the Genome Institute of Singapore because right now I have an internship. It's just that I perform certain experiments in my lab because it's more convenient. It's like a kitchen, you just know where things are, where's sugar, where's flour, and so on and so forth. But on a daily basis, because I have 2-3 major projects, and this project, for instance, the project on splicing, is purely computational as of now. So basically just machine learning and bioinformatics. And for this one, I don't even have to come to NTU. So the bulk of the work, I actually have done it at home last year.
Okay.
And that was quite convenient, also because it was post-CB and everyone got used to this lifestyle of just staying at home and doing stuff.
CB means what?
Circuit breaker.
Right.
Okay.
Yeah, semi-lockdown we had. Yeah.
Yeah.
Because people in other parts of the world will be listening and wondering, what is CB?
Oh, yeah, right. This is Singaporean context.
Yeah.
Right.
Yeah, so right now I'm actually doing experiments for a different project, which is for the internship at the Genome Institute. I come to the lab quite late, like 11 p.m. perhaps, and then I...
11 p.m. or a.m.?
p.m. Oh, sorry, a.m. Of course, a.m.
Right.
No, sometimes I do come at 11 p.m.
Okay.
Yeah,
Okay.
there are days when I'm staying till 3 a.m., 5 a.m.
Oh wow.
But this is rare when I'm pushing something really hard.
But what is it that's keeping you that late? Like.
It's just because sometimes you get frustrated that you can't get a certain process finished. Because in biochemistry, because our lab is basically biophysics and biochemistry, this is a wet lab. So there are in SPMS we have wet labs and dry labs. Should I call them that? So basically, wet lab implies that you're working with chemicals.
Right.
And it looks like we are just pipetting very small amounts of transparent liquids the whole day and testing the result.
Yeah, I'm wondering actually, how does DNA look like? Like, what sort of thing can you, that we know of, that we can relate with? Does it look like soap? Does it look like... Because it's material, right? When you're talking about splicing, it's not like you actually go and do it. You mix fluids, I suppose, right?
Yes, for splicing, because this is computational, but yeah, for our other projects, I'm actually working with purified DNA and RNA. And if you dry it out, it's possible to dehydrate it. Especially for DNA, you can actually dehydrate it, and it will just look like salt residue on the bottom of your tube. So, just like a spot of salt. So it's basically just white, non-transparent thing. And interestingly, DNA is very, very stable, so you can rehydrate it at any moment and it will be intact. Same DNA. So you can actually dehydrate DNA and send it over mail to the other part of the world. That's totally possible. And it will be safe and intact, which is really nice.
Interesting.
So in the looks, it's not super unique. As in, if I were to look at it, I guess it just looks like...
Yeah, it doesn't look anything special. Unless you
not
have
like it's
certain...
coding for life.
Yep. Unless you have certain ways to look at it, which I'm actually working on. Yes.
Are you working? So you're working on, developing ways of looking at the DNA, or
not exactly, but I'm using it. I'm developing one method to look at the secondary structures of DNA.
Oh.
I can explain a bit more about it.
Of course.
I think.
So one of my projects, the one that I'm working on right now, is involving nanopore sequencing and wet lab experiments with DNA and RNA. So mostly RNA, actually. So nanopore sequencing is a relatively novel technique of reading the sequence of DNA or RNA. So basically DNA and RNA, as you probably know, is just a polymer consisting of nucleotides decoded as letters, right? A, T, C, G. And so there are techniques, because if you don't know what the sequence is, you can't really bioinformatically analyze the gene, right? Because the bioinformatical analysis of the gene is a very important part of any personalized medicine and so on and so forth. Because if you have some mutations, then it might be critical. There might be actually a personalized drug development and so on and so forth. So nanopore sequencing is one of the techniques to actually read the letters that are comprising a human genome or any other genome or any synthetic DNA sequence or RNA sequence. And this is what I'm working on. It is novel, and the technique itself is quite cheap. So it's commercial. It's the company is called Oxford Nanopore, and I think it's quite a successful company these days. I think they are included in the most successful MIT technology companies or something. There's a digest like that, right?
Okay.
And so the technique itself, you have an array of nanopores. You can just buy their device, which is smaller than a phone and is cheaper than the latest iPhone, actually. So, and this device has an array of nanopores, and then you have a specific set of buffers and other chemicals, and you mix your DNA of interest, the purified DNA or RNA of interest, with all these buffers, etc. And then basically the system itself is biological nanopores covered with buffer which contains ions. And then you apply voltage, and the nanopores are injected into a membrane, a solid state membrane. And then you apply voltage across this membrane, and the ions from the buffer start flowing through the nanopore. And you can actually measure this current. So this is precision engineering. This is like tiny currents, like picoamperes or something like that. You can still measure it. But then if you have DNA or RNA in the same solution, because it's negatively charged, because the backbone of DNA is negatively charged, it will also flow through the nanopores. And you can detect the changes that DNA or RNA nucleotides are causing in this current. So basically, a flow of ions, and ions are small molecules, right? So there will be a lot of them going through the nanopore. And then you have a relatively large DNA molecule going through, and you can actually detect, okay, right now it's A going through the nanopore, now it's T, now it's G, and so on and so forth.
That's quite interesting.
So are you able to detect to that level of resolution, or you detect a number of them at once?
You detect the sequence, the letter that's going through. So
Each
we basically, we already know what's the signature of every nucleotide, and that's how we read them. It's not super precise. The error rate is still about, I think, 5%, but because you also read a lot of molecules at once, same molecules, right? And then you just align them, and it reduces the error. So this is very interesting. But I'm using this technique not to read the sequence itself, but to try to detect secondary structures in DNA, which is a completely different thing.
What are secondary structures?
Yes, so basically, so when you think of DNA, you probably think of this ubiquitous double helix, right?
Yes.
So this is, actually DNA is in this double helix state most of the time because it's very energetically favorable, it's easily packed, and so on and so forth. But in certain transition processes in cells, like when the cell divides or when it's transcribed into RNA and so on and so forth, you will have DNA unfolded, and then it might form other structures, secondary structures.
I see.
And initially we thought of them as of just, exotic structures, nothing interesting, but it turns out that these structures are actually regulatory mechanisms in cells in their own way. So basically, if there is a certain sequence, it can fold a secondary structure, a particular secondary structure, and we're studying one of those, which is quite famous these days. It's called a G quadruplex. So basically, it will just form stacks of guanines, planar stacks of guanines, quite a peculiar structure, very recognizable. And this structure can actually affect the cellular processes as well. So basically, there are certain proteins that will recognize this particular structure, and then it will affect the downstream processes like transcription, translation, etc.
So can they fold in such a way that they hide some things?
Yes, exactly. They can.
And so in that way, they regulate that part.
Yes, precisely. They can hide the gene promoter regions. So basically, gene promoters are the keys to recognize for the protein to recognize that this is a start of the gene. So we need to start transcribing here, so it can hide it.
And once it's hidden, then you can no longer get that gene out or expressed.
Exactly.
So,
and there are drugs stabilizing these secondary structures.
So how's the problem tackling process in your research? Like, do you work in groups or do you work individually? And then what's the thinking process towards reaching a goal that you set out?
It really depends. It's a really complex system. Mostly I'm working alone on a daily basis. Sometimes, because I'm a theoretical physicist, so I don't really have good training in biochemistry or wet lab training. So I definitely need to ask some advice and guidance from the members of our lab, which are amazing and have amazing skills in these things. But mostly it's just that we have a brainstorming process in the beginning. But usually we don't even have a precise question that we want to ask. So it's more like some discovery in the process. We have a technique, we have an idea that there might be something interesting. And then usually my supervisor just gives me some freedom to explore it. And then if it works out, if there is something interesting indeed, then we pursue it and then we push it and try to work on it harder. Sometimes there's nothing, but usually there is at least something interesting.
I see.
Just a quick question. You said nanopore sequencing. Is that different from DNA sequencing? Is that just a process, or is
nanopore sequencing is a type of DNA sequencing. It's not the only type, so there are different techniques, but I'm working on this one.
Okay. So I've heard that it used to be very expensive to sequence DNA, and now it's far, far cheaper. Can I, as an individual today, as in what kind of skills are really required to do your kind of work? Can I just go into my room, get the right tools for $200 or less, and be able to to sequence DNA?
Actually,
is that the state of things now?
Actually, with this nanopore sequencing, theoretically you can. It will be slightly more expensive than $200, but initially, I think in 2007, it was about $1 million or $1 billion. Right now, you can just buy this device. And because it's very down to earth, because it's developed for people to use it in field conditions, like anywhere. I think it was even used in the International Space Station with very minimal training. So yes, these days you can. You just need to extract some DNA from yourself, and then just need some pipettes and some pure water, and all the reagents are provided. So you can just buy it. It will take a while and some processing, but it is possible these days.
Okay, so yeah, something you mentioned earlier, like when you're not working in the lab, right? You're just working with data. So what is this data, and did you collect the data, or is it already available somewhere else?
For the project that I was working on previously, so sometimes it is my data collected, for instance, for the nanopore sequencing experiment, because there's also quite a lot of data processing. But for the other project, it's mostly pre-collected data, because actually these type of projects which involve machine learning, I think it's really cool that these days you can take advantage of the big data available, because there are these huge data sets published quite recently in the last few years. And these data sets collect RNA from many humans in many, many conditions. There are thousands of them, tens of thousands. And it's amazing, because we actually took advantage of this in our recent project. And we're trying to study it on a big data level, which I think is amazing.
So then what are the functions of these, I'm going to say neural networks, because you're probably working with neural networks when you say machine learning. So what are the goals that you set for the network? What, like the output?
So the goal is to predict alternative splicing. So basically our initial data set is just inputs of sequence, which is the same, plus auxiliary signals from the cell, which we discussed, right? So the protein levels and so on and so forth. Protein levels, just protein levels. And then, so we have a training set of, let's say, 16 tissues and then 250 patients for each tissue. And then this is quite a large data set, right? So we're hoping to extract as many correlations and interdependencies from it. And now when the network is trained, so for this type of network, it actually takes a while to train it. It took us 2 or 3 weeks on a cluster.
Wow.
Yeah, so, and now we want to predict. So basically we have a new sample, right? And we have these auxiliary protein levels. And now we want to know how these particular gene will be spliced. So basically how this particular gene, in which protein it will result afterwards. This might be quite powerful.
And how are the preliminary results so far?
Well,
Are they
it seems
promising?
it's quite promising in terms of, in data science and machine learning. In this field, people usually care about correlations and pure numbers. So basically, if your benchmark test data set is the performance of your neural network on your benchmark data set is 0.85 instead of 0.81, that's amazing.
Right.
That's a breakthrough. I mean, obviously for biology, so in this sense, our network is performing quite well compared to the previous one, actually much better. But for biologists and clinicians, this is not very significant, right? So this is still on the data level. So we do extract some features. It means that we do extract something meaningful, but that's why we need to outline a very narrow example of how we can apply it.
Okay.
And that's what we're working on right now.
And you said you were trained as a theoretical physicist. So how was getting into machine learning? How did you find the journey?
Well, I would say that actually getting into machine learning was much easier because theoretical physics is practically mathematics and machine learning is practically mathematics too. The hard part for me is getting into biochemistry and experiments. This is something I'm still not struggling, but sometimes I'm experiencing problems with that because as a theoretical physicist, I think I have this mindset of if everything works perfectly on paper, then everything should work perfectly in the lab. And in the lab, I always underestimate the complexity because really you perform the same experiment and the result is not always different, but sometimes it's different and you don't really know why. You do exactly the same thing, and maybe you've heard about the crisis of reproducibility in biology and medicine, because the more complex the system, the harder it is to reproduce the results of the experiment.
There's so many variables that you cannot account for.
Exactly. Like your pipetting style is slightly different from your colleague, and the result will be different. Seriously.
Yeah.
So just to clarify, you say that when you are training your machine, you give it some gene, and then you give it some
additional signals,
and then you want to predict what would be the final protein that's produced. I see. Okay, so this was more specific to your work. Now we want to move on to something a bit more general about DNA. So the common person, when we think about DNA, we think about the double helix, because that's what we see a lot more. We hear about the discovery of DNA in the last century. Well, could you give us a sense of what are the biggest breakthroughs recently in this field, in the field of whether it's DNA editing or DNA engineering in general?
So that's a very interesting thing to talk about. So because I'm not really working on bioengineering techniques like CRISPR, for instance, I would love to tell you more, but I'm not an expert in that. But what I can say is that previously in DNA we thought, maybe you know that only a very small part of DNA, of human genome is actually used as genes, is transcribed into RNA and proteins. So basically around 1, if I'm not mistaken, or 3% of DNA is genes. Everything else is the so-called junk DNA. And
that's what we thought.
That's what we thought before. Maybe 20 years ago or 30 years ago, it was still a common notion. But right now, we know that actually this DNA is not junk DNA, and we're just starting to discover all the motifs that are present in these regions, which are not genes, and how important they are. And this is partially what we're working on as well. This is pure bioinformatics. And can you imagine, because the length of the human genome is around 3 gigabytes, so it's very, very long. And the genes are only 1%. But all the other sequences, some of them actually carry certain motifs that might be important.
I, in fact, want to ask about CRISPR.
Right.
I'm interested in understanding what is it that can happen if CRISPR were coupled with machine learning, because the way that I am thinking about it is that if you can, I guess from what I understand is that with CRISPR you can essentially cut out things, right? And therefore you can create new gene sequences, you can create new
Right.
forms of proteins that perhaps don't
Right,
exist in
right,
nature.
right.
And with machine learning, you can then perhaps, before you do that, you can perhaps predict what will this thing do in
Exactly.
the natural environment.
Exactly.
So I was wondering if there is any research going on in that area, coupling these two things in terms of predicting maybe a protein, and then on a high level predicting an entire organism. Like if I get the
Right.
sequence of
Right.
genes, would it code for this kind of organism?
So this is all very advanced, but this is definitely a direction we're heading towards, because indeed the CRISPR technique is very, very powerful, but probably it's too early to apply it because we don't really know, because it's also correlated with these motifs in DNA, right? You can cut off a certain motif thinking that it's not important, but it turns out to be important. That's why the machine learning can actually play a huge role in this. So if we can predict what exactly will be the result of cutting off this part of the genome or inserting something using machine learning techniques, then it will be amazing. But the problem here is that for a good machine learning model, you need a training data set. Right. So it means that you need, how can you predict something if you don't have any data on which you can train the neural network, right? So basically, in order to predict these patterns, you will have to perform a lot of CRISPR experiments, and probably on organisms that are as close to humans as possible, and then collect the results as precisely as possible on each possible level, like molecule, like protein expression, and maybe some small molecules, and so on and so forth, in each tissue type, in each cell, right? And then you can train a neural network, and then you can predict it. But can you imagine the amount of work that you need to put into it? So I think we're heading towards it, but maybe we'll reach these capacities only in, I don't know, 50 years, 80 years?
Maybe 10 years. That's always 10 years.
It's always 10 years.
But what you're saying is that we are lacking the data for us to train the machines, and once we have sufficient data, then perhaps the machine can help us. So AI algorithms can help us make predictions for what sorts of whether. I'm interested in really, is it possible to create completely new life? Because the things that people talk about is that, one, you can create new alphabets, it appears. Not just ATCG, but you can have other alphabets. And I'm wondering, that
is
seems
true.
To be very
that is true,
now.
But the problem is that this alphabet is perfectly optimized by million years of evolution.
You mean the one we have?
The one that we have. It all comes down to the first organic molecules, right? And inorganic molecules as well. So the problem is that ATCG, you can have some DNA designer nucleotides, but probably the chemistry will be really difficult to optimize. Maybe you can do some basic stuff, but because of the atmosphere of the Earth and all of the carbon chemistry that we have around us, it's so optimized that it will be enormous effort to create something new. But it's possible.
It's possible. So there's the sense in which we know what is possible and what is allowed. Perhaps I'm wondering what is ethical, for example, in whether it's DNA editing or in bioengineering in general, and then there's what is allowed and what is possible, right? In your mind, what do you think is possible, say, presently, as well as looking ahead next 5, 10 years?
I think editing of the embryonic materials is possible, and we're probably heading towards it. I think it's probably the only application that's going to be that we're going to see in the next 15 years, or maybe slightly later even, because otherwise it's really difficult to edit something in the adult stage. So I think we definitely need to start with embryonic material, and maybe from some simpler organisms first, maybe from animals. But for now, it's completely unethical because we have no idea how it affects the downstream processes.
And the only way to know is that we have to do it, right? That's what you said earlier about the data. So
Exactly.
Someone has to. We can, even if we wait for 50 years, we won't just have the data. Someone has to.
Well, unless we build a perfect model, like from the quantum physics level, we build a perfect model that is describing human body and the behavior, which is kind of not very likely for now as well.
Right.
Although possible. That's another approach, right?
Right. So I'm thinking that some things we can delay because we don't feel comfortable with it. I mean, obviously, like you mentioned, editing embryos, or, I mean, as people are saying, design of babies per se, and
Right.
trying to predict how the human will behave after you change some things. We'll never probably get it right until we have the actual data, unless someone actually goes through it.
Of course.
So.
But now at least we have 1 or 2 babies from China, right, that were edited at the embryonic stage.
Right. And those were, I guess, for those, maybe you want to tell people, for those who don't know about that.
Yeah, I don't. Okay, so as far as I remember, there was this Chinese scientist who, against all ethical laws, edited the genomes of the embryons of 1 or 2 Chinese babies. I'm not actually sure. I think it was 1, 2. Yeah, because these were twins, right? So, and because it's forbidden in every country in the world, and he wanted to protect them against HIV, because their mother was sick with HIV. She was infected with HIV, and basically he wanted to edit this gene that can help with that, because apparently there are a few genes that are correlated with anti-HIV effects in humans, because most of the humans are not immune to HIV, but there's a couple of gene mutations that can actually affect your immunity, and apparently he recreated this mutation in order to create an immunity for HIV. It was not completely ethical, also given that it was not a life-threatening condition, because with all the modern medicine, likely they would not even get HIV in the first place.
Right.
Because apparently their mother was also on the antiretroviral therapy, which makes it a little bit pointless. That's why it's twice as unethical. But we'll see the result in 10 or 20 years.
Right.
So that's one data point. So we hope we hope that it will be useful in some sense in the future.
Yes, but we need thousands and tens of thousands.
Okay, I think my last question on this speculation front, I have seen a few ads where people are saying that we can tailor an experience to your DNA. For example, some people think that your eating routine or your exercise routine and your, I don't know, anything to do with your life can be tailored depending on your DNA.
Right.
How far should we trust this sort of advice? Is it too complex? Are we too complex for those kinds of things, or can there really be legitimate recommendations that suit us based on our DNAs?
Yeah, so I think you actually, judging from how you asked this question, you already know it better than me. But yeah, I also took the DNA test to get all these recommendations, but mostly for diseases and everything for mutations. Indeed, to some extent, for certain very particular things like celiac disease, for instance, it is possible. Or it's possible to predict a likelihood of certain cancer, for instance, but it only works for a few diseases, a few cancers, a few recommendations. Everything else is more of a speculation, because human body is highly adaptable. So basically, any condition you put us in, we will adapt in a certain way after a certain period of time. So any diet that you have, unless it's really contradictory to one of your mutations in certain genes, right, it should be okay. So for instance, lactose intolerance might be quite serious, but everything else, alcohol intolerance might be quite serious. But everything else is very, very flexible, because humans have survived on very scarce diets for very long periods of time, right? And actually in certain regions, for instance, I read about it, and I'm not sure how speculative it is, but apparently in certain regions of China or Mongolia or even Russia, people have really scarce diets and they still have a very long life expectancy. So we're not sure, because the body is able to produce certain peptides, let's say, from other available materials in the body. And all these diet recommendations are just a very, very highly, it's a top layer of many, many layers of processing, and we actually don't know what's going on in between. So it doesn't really make sense to listen to these recommendations that far. It is highly, highly complex. Certain things, but even for cancers, you can predict breast cancer, for instance, because there are a few mutations that will lead to breast cancer with a very high probability, these BRCA genes. So for instance, if you have 2 of the mutations, you will develop a breast cancer with a probability of 80% or something like that. But everything else, even for other types of cancer, some mutations might increase the probability, but not as significantly. So even that is not that fatal.
So generally, what you're thinking about life, how has it changed since you have an understanding of life that's probably different than common person on the street, I suppose.
Right.
You know, to understand what really makes us grow, how do we convert food to what we are. Do you think that has affected how you view life, or how you view the world how you view life in particular? Has that affected you?
I personally think that it did not, but my friends tell me that, especially friends outside of science and academia, do tell me that I take the world and all the information very rationally, and sometimes it gets in the way of just, enjoying the life. But I can't quite revert this process. Maybe I can just learn to, live with it. But it doesn't bother me because I enjoy it, because I think it's actually very enriching to know how things work, because it doesn't simplify the world for me. It doesn't make it less enjoyable and mysterious and romantic. It actually makes it more enriched with information and with dependencies. I think it's very, very interesting, because the more you know, the more dependencies you discover, right? And this is still very mysterious. So I actually like it. Okay.
So you think about life in very mechanical terms, I suppose.
I think so. That's what my friends tell me.
It's understandable from a physics point of view.
All right. So at the end of each episode, we'd like to stray away a little bit from the research and talk about your personal interests, things you do in your free time, things that you do to
unwind.
Yeah, unwind basically. So can you tell us a little bit about that?
Sure. Right now, maybe is not the best time to describe my hobbies, because I'm a last year Ph.D. student, so most of my hobbies
a lot of research.
just reading papers these days. And also, I don't really have many active hobbies like many people do. I just do some sports, but basically just gym, so nothing interesting. And sometimes I pick one thing or another. Like I used to do vocal and drumming, but right now I'm not doing any of this.
Were you doing them on your own, or did you take lessons?
I took lessons for both.
It
was quite fun. Right now, because right now I'm watching quite a lot of anime, surprisingly, and I'm thinking to start learning Japanese.
Nice.
But for some reason with languages, I'm not sure how it works for other people, but I feel that any language you start to learn, as far as you're not forced to learn it in terms of environment, if you're not actively put in this environment, it's really difficult to invest into it. So any investment into any language apart from English kind of doesn't pay off. I used to know French quite well. Right now, I just forgot it.
Okay. Now, just out of curiosity, how many languages do you speak right now?
Well, I can safely say that I speak 2 languages. My native language, Russian, and English. I used to speak French relatively well, but now I just remember some poetry and songs, that's it. I can read it still.
So music, languages, and a little bit of sports.
Mostly. I listen to a lot of music. I'm quite a music head, but this is not an active hobby as well, right? I do draw sometimes, though. I enjoy it.
Okay. That's cool. And what do you think about life in NTU? Because I'm assuming you spent some time living on campus when you came here.
Right.
So what was your impression about the facilities and the student life?
Actually, I was quite impressed when I just came to NTU, because I was living on campus for 3 years. And I can't say that I used the facilities that much, but I did enjoy those that I used actively. For instance, we have a really nice pool.
Yes.
I think it's really nice to relax and unwind over the weekend. And also because most people for some reason are afraid of the burning sun during the most active hours of the sunlight ray. So usually it's kind of empty during these hours. Also not good for your skin, but yeah, I'm Russian, so I miss the sun. I'm tired of cold. And everything else, I think it's really nice to just walk around because it's so far from everything. And the atmosphere, I think it has a certain kampong spirit.
Right.
And
what is kampong spirit?
Kampong spirit is this is a Malay word, if I'm not mistaken, right? Used in Singapore. So kampong means a village. So kampong spirit means village spirit, and there are a few places in Singapore that still have it, like Pulau Ubin, for instance. This island, this small island with a few landed properties, and there are chickens walking around and so on. I actually love to visit this place. This is one of the
it's
wildest places
in
Singapore.
It makes you forget that the city and the all the noise of the city and all that. Yeah.
Yes.
That is weird. People are fighting tooth and nail to get out of villages, and then you want
That
to
is
go
true.
to a village kind of life.
That is true. When I was living in my hometown, I wanted to get into the big city. But now, especially because we're not traveling for more than a year, I really miss this feeling of being in the wild, because we're constantly in this city jungle. I'm not sure.
Right.
Do you miss it?
Yeah, of course. Yeah. I mean, I also love going to Pulau Ubin. It's just that just picking up a cycle and then just going around and then resting in the different spots and just contemplating, the views and it's nice.
It's a lovely place.
Last thought or question for me. I want to go just a little bit to languages. Do you see any similarities between the human languages and the DNA language? Because it is a language as well, right?
It is a
grammar,
language, but it is very, very different. I never thought about it, actually. This is a very interesting thing. Maybe we can write a machine learning program and neural network that will transcribe the DNA language into some sort of music or something, because there are definitely patterns in it. It's just that I never thought about it in this way. Because it's just one sequence, right? Maybe I should work on it. That's my next project. I promise.
Okay. Well, I think that really
wraps it up
for us. Anna, we really appreciate the time we've spent with you and the things you've shared with us. And we hope you enjoyed. Really, we are doing this because we believe that people at NTU are doing really interesting research, and they never quite get the time to share with the community and the world. And so through these, we hope that we can get more people to casually talk about their research. So thank you, Anna. And
thank you for having me. I was happy to share and would happy to come again. Would be happy to come again.
Yeah, doors always open. And thank you.
Thank you.
Thank you. Bye-bye.
Bye-bye.